Hey guys, welcome back. Michelle Glass here, and we are still talking about special senses. And our topic for today's video is hearing. And hearing is happening in the part of the inner ear called the cochlea. So we're going to be investigating that in a little bit more detail as we move along. Okay, so let's start by kind of drawing out our diagram of the ear. We're going to be super simplistic with our diagram, so you should be able to sort of replicate this. Of course, you have been or will be studying the anatomy of the ear as part of your lab work, so this should be making some sense to you. Let's start with our external ear. Of course, I left off my pinna or auricle, the part that's going to funnel sound waves into my canal. I've listed my canal here as the external auditory canal. Sometimes you'll see it listed as acoustic meatus, which means the same thing, just sounds fancier. Sound wave is going to hit up against the tympanic membrane, which you called your eardrum when you were not an anatomist, but now you are, so make sure you're using the correct terminology. Okay, from the tympanic membrane, we're going to see that the sound waves are going to be transferred through three little tiny bones in the middle ear. This is actually in that petrous part of your temporal bone, if you recall that from your study of the skull. You have first your malleus, which will knock against your incus, which then knocks against the stapes. So these are your three auditory ossicles. This is the section which is called the middle ear. And then I've just drawn my cochlea. So I've not drawn my whole internal ear structure, just the part that we really care about right now in our discussion of hearing. Notice that the stapes is knocking against the cochlea, specifically at a structure called the oval window. So that's a part where there's not bone right there. So that's going to be transferring the sound waves into pressure waves in the fluid that's inside the cochlea, specifically the perilymph. And then we see that those waves have to have a way of dissipating, and they're dissipating at that structure called the round window. Now, the cochlea is the part that contains our, the part of our ear that's involved in hearing. You will see the structure called the spiral organ, or you might see it listed as organ of Corti, which is how I learned it. The spiral organ is located inside the cochlea. It is the spiral organ that's going to contain our hair cells, which are our Meccano Receptor cells. So we have studied olfaction and gustation, both of which contained chemoreceptors, receptor cells that are stimulated by chemicals binding. Now we're looking at what's called a mechanoreceptor, and this means there's going to be a mechanical movement that will actually stimulate the cell. And these cells are called hair cells. We'll see hair cells in all of the structures and sensations that are detected by the internal or inner ear. And what we'll find, and what you may already know from your lab study or from other study, there are really, we can think of like three sensations being detected by the inner ear. And the topic for this video, of course, is hearing. So that's what we're going to focus in on. So again, the inner ear, it consists of three distinct structures: the vestibule, the semicircular canals, and the cochlea. And the vestibule and the semicircular canals are both involved in detecting that sensation of equilibrium. So that's our next video presentation. The cochlea is, of course, what we're focusing in on here, because this is, again, where hearing is detected. Now, the structure of the inner ear is the same for all three, meaning it's organized by having what's called the bony labyrinth. So there's actually bone wrapped around the outside. And then in the middle of that bone, you have a membranous labyrinth. Now, there is a space in between the bony labyrinth and the membranous labyrinth, and that space is full of fluid. That fluid is called perilymph. So lymph is meaning fluid, and peri means around. So perilymph is around the membranous labyrinth. And inside the membranous labyrinth, you have fluid, and that fluid is called endolymph because it's the innermost fluid, okay? Now you're gonna see that it's movement in the perilymph that will actually stimulate movement in our hair cells. That's our Meccano Receptor cell, so that movement then is gonna stimulate our receptor cell, which then synapses and stimulates our sensory neurons. Now that membranous labyrinth is extending through the vestibule, semicircular canals, and the cochlea. And as we are reading about the part of the membranous labyrinth that extends through the cochlea is called the cochlear duct. So notice I've drawn a spiral here for my cochlear duct. If we go back to my beautiful drawing here of my internal ear, you can see that my cochlea has been drawn as a spiral shape. So it's a spiral shaped structure. Think of a snail shell as probably a good reference, okay? So I'm gonna take that snail shell and I'm gonna unwind it a little bit. So I'm gonna unroll it in my diagram here. So on the outside here, this would be the bony labyrinth. This here, this blue is my membranous labyrinth, and remember we're going to call it the cochlear duct once we're inside the cochlea. Now you're going to see that there's two spaces on either side of the cochlear duct. You have from the oval window the vestibular duct, so sound wave is entering here. Sound wave is turned into a pressure wave and the perilymph fluid because you had, again, that stapes knocking up against the oval window. And then that fluid is going to move through the cochlea. Depending on its frequency, it's going to cause movement in the hair cells of the cochlear duct, and then really hair cells of the spiral organ. And then we'll see those pressure waves dissipating through the tympanic duct. So it's tympanic duct because we're heading back towards the tympanic membrane and dissipating at that round window. So think about your ocean having a tide that goes out and then comes back in. So it has to have this ability to go out through the perilymph and then go back in at that round window. Now if we go back to our diagram here, we can also talk a little bit about the significance of the auditory ossicles. So we're taking this, a big membrane, tympanic membrane that's doing lots of vibration, and then we're transmitting that to these smaller and smaller and smaller bones so that that stapes is really knocking up against the oval window. So we're taking this big tympanic membrane and then knocking it up against a smaller space. We're really amplifying those sound waves as we turn them into these pressure fluid waves in the perilymph of the cochlea, okay? All right, so what we want to look at next is really what's happening here. So I've said movement of the perilymph is going to cause movement of the hair cells at the spiral organ. So what does that actually look like? So that's what we want to take a look at next. Okay, so let me move my picture. I'm trying to draw, like, we're zooming into this area here. Oh, going back. Okay, let's see if I can. Okay, so my idea here is you have your vestibular duct, which is full of your perilymph, and then you have your tympanic duct, which is full of your perilymph. And then you have in the center here, you have your cochlear duct, right? And it's full of endolymph. And you also have kind of, lining this area here, you have what's called the spiral organ. Okay? The spiral organ, or organ of Corti. Okay. Now notice you have a tectoral membrane that's sitting on top, and it's not touching your epithelial cells here. These are hair cells. These hair cells have stereocilia sticking up, which we're going to be talking about more in a minute. And then we have at the bottom the basilar membrane. You see it? Here's my super zoomed in picture of my spiral organ. So again, oops, tectoral membrane on the top, basilar membrane at the base, and here are my mechanoreceptors, my hair cells. My mechanoreceptors have those cilia sticking up. They're called stereocilia. Notice you have a tall one and they get shorter and shorter and shorter as they go down. So this really allows for some sense of directionality. Notice that these cells are synapsing onto sensory neurons. So we're going to see that movement of the perilymph is going to cause some movement, some bending of our stereocilia, which then gets transmitted to the sensory neuron. So this is what's going to happen. As you get those sound waves beating up against the tympanic membrane, transmitted to the malleus, incus, and stapes knocking up against the oval window, you get that sound wave turned into a pressure wave in the perilymph. That perilymph is moving through that vestibular duct into that tympanic duct back towards the round window, right? And as it does that movement toward the round window, it's going to hit the basilar membrane. So the basilar membrane is moving, causing those hair cells to bump into that tectoral membrane. So when those stereocilia get bumped against the tectoral membrane, then they move. That's our mechanoreception. That movement stimulates the cell, which in turn stimulates the sensory neuron. So that's how we detect sound. Now what we'll see is that frequency is detected by what part of the basilar membrane is moving. So it's going to be throughout that whole spiral organ. You're going to see, if it's a low frequency, it has a very long wavelength, and so it's going to stimulate that basilar membrane, further along the spiral organ than a real squeaky, high-pitched sound, which is going to have a shorter wavelength, and it's going to be hitting that basilar membrane closer to kind of that entry point. So frequency is detected by what part of the spiral organ is being stimulated, what part those pressure waves, how far those pressure waves reach, and then that section of the spiral organ is stimulated. Volume is more like let me rephrase. Volume is the activity and the number of hair cells involved. So a really loud sound is going to be hitting harder and more cells are involved than a super quiet sound. Okay? And so it makes sense where if you are exposed to the same frequency at very high levels, you're going to have damage to those hair cells and have some hearing loss in that particular area, but maybe not universal hearing loss. Okay? All right, so let's diagram our pathway. Sound waves are entering that external ear. So they're being collected by the pinna or auricle, traveling through that external auditory canal and hitting up against the tympanic membrane. Once that tympanic membrane vibrates, it's transmitting that vibration to those auditory ossicles, malleus, incus, stapes. The stapes hits against the oval window, transmitting the sound wave into a pressure wave in the perilymph of that vestibular duct. Okay, and so then depending on frequency, it's going to move, if it's a low frequency, it's going to move further down the vestibular duct. If it's a high frequency, it's not going to move as far. And so then we're going to see that we're going to get vibration of the basilar membrane as that perilymph wave is moving through that tympanic duct. The basilar membrane is going to cause those hair cells to bump up against the tectoral membrane. And so then this is going to cause those stereocilia to move, and that's going to open up ion channels. So now we've depolarized our Meccano receptor, which means we're going to be sending that signal onto our sensory neurons. Those neurons are going to make up part of the cochlear nerve, and the cochlear nerve is going to form the vestibulocochlear nerve, which is cranial nerve 8, which you might have also called auditory nerve. Both names are equally correct. Now you can understand as we progress through why it's called the vestibulocochlear nerve. Okay, and then we'll see that those signals make their way to the brainstem, the thalamus, which is, remember, our relay center. We're going to see that pattern the rest of our conversations here. And then that sound wave information is processed in the auditory cortex, which is found in the temporal lobe.